Thanks, everyone. And I'm real pleased to be with you today to present one of my paper. So let's get started. So globally, around 11% of infants are born prematurely, kind of 6% in Norway. Those premature babies have high risk of infection and inflammation. There are many reasons behind that, but I'm going to mention only a few of them. They have immature immune system. Also, they have impaired barrier function. So the gut microbiota could be transferred from the gut to blood circulation and cause sepsis. Also, the preterm babies have high abundance of pathogenic bacteria and low abundance of commensals bacteria. Besides, those preterm babies have lower microbiota complexity as compared to the full-term neonates. So that's why those preterm babies have high risk of infection and inflammation. So how we tackle this problem? Simply by giving them some antibiotics. That's why kind of 80% of infants in intensive care units receive antibiotics to protect them from infection and inflammation. However, the antibiotic treatment actually exacerbates the case by killing the commensals bacteria and subsequently increase the abundance of opportunistic or pathogenic bacteria. That's why in many countries, including Norway, probiotics have become a standard for reducing the GIT inflammation and infection. However, this result actually is based on empirical evidence. So our objective was to evaluate the influence of probiotics on extremely preterm infants' gut microbiota, resistome, and mobilome. That's why we designed our methodology as follows. We recruited 3 different categories of infants: extremely preterm babies with gestational age 26 weeks, very preterm babies with gestational age 29 and 30 weeks, and full-term healthy neonates with gestational age 40 weeks. The extremely preterm babies received antibiotics and probiotics, while the first subset of the very preterm babies received antibiotics, while the rest of the group received none. Please keep in mind that the extremely preterm babies received antibiotics for longer period of time as compared to the VP1 group, 9 days versus 4 days. Then the fecal samples were collected at 4 different time points, and then the DNA extraction, shotgun sequencing, and bioinformatics analysis were performed. So let's talk briefly about the microbiota composition then. Well, the domination of the genera here in the probiotic-supplemented group was for Bifidobacterium. And the relative abundance of Bifidobacterium here in this group was quite comparable to the full-term healthy babies. But if we compare that to the non-probiotic supplemented group, we can see here that the domination of the genera in those groups was for some opportunistic or pathogenic bacteria, like Escherichia, Staphylococcus, and Klebsiella. So this suggests actually that the probiotic use can restore the normal infant colonizer right away after the treatment. Also, if you can compare the antibiotic supplemented group and non-antibiotic treated group, we cannot see here a huge difference between those groups. And this actually suggests that the antibiotic treatment has low or negligible impact on gut microbiota in early life. However, this also can suggest something very important, that the hospitalization itself can promote the infant's gut microbiota with some opportunistic or pathogenic bacteria. But yeah, after discharge from the hospital, those preterm babies can restore some commensals gut microbiota. So then let's talk about the microbiota maturation. We performed the Richlet multinomial mixture, which is one of the machine learning technique to cluster the infant's gut microbiota community types into different clusters. In our case here, we have 5 different clusters. MC1, the least matured microbiota community, which was dominated by Staphylococcus family. While the MC5 which was the most matured microbiota community, which was enriched in Bacteroides and Bifidobacteria. Also, please keep in mind that MC4 is the second highest matured microbiota community, which was enriched in Bifidobacteria. So the domination of the microbiota community type in the probiotic supplemented group was for MC4. And also the proportion here can be comparable to the full-term healthy baby. But if we compare that to the non-probiotic supplemented group, we can see here the domination of the microbiome community types was for MC1 and MC2, which were dominated by some opportunistic and pathogenic bacteria. So we can, from this result, suggest that the probiotics might promote the microbiota maturation. But to be a bit sure about that, we ran random forest modeling, and we can see here that the probiotics among the top 3 predictors of mature gut microbiota. Then to gut resistome. The relative abundance of antibiotic-resistant genes identified in the probiotic supplemented group was significantly lower than those identified for the non-probiotic supplemented group. And here as well, the relative abundance of antibiotic-resistant genes in the extremely preterm babies was quite similar to the full-term healthy babies. So actually, this suggests that the probiotics can alleviate the resistome burden. But here as well, we don't see here a statistically significant difference between the antibiotic treated group and non-antibiotic treated group. So also this supports the same idea I mentioned, that the antibiotic itself has maybe low or negligible impact on resistome in early life, but also because of this group has higher relative abundance of antibiotic-resistant genes, this supports that the hospitalization might increase the antibiotic-resistant genes abundance. But for the rest of the time points, we didn't see a statistically significant difference between groups. To gut mobilome. Here as well, the relative abundance of mobile genetic elements identified in the probiotic supplemented group was significantly higher than those identified for the other groups. And we can see here the same trend across the study, except for the third time point. And this actually made us wondering whether the probiotics could be a factor for enhancing the long-term horizontal gene transfer. That's why we decided to sequence the Infloran, the probiotics product that was given to the extremely preterm babies. Here we found some mobile genetic elements encoded in the probiotic strain genome. And this actually made us wondering, what if the probiotic itself transfer those mobile genetic elements to some opportunistic or pathogenic bacteria? Will the antibiotic-resistant gene dissemination rate increase? This is interesting. Also here, the mobile genetic elements identified in the probiotic product was less than 1%. So it's not very high. But actually, this is very interesting result, but actually merits further analysis. Let's talk about the co-occurrence analysis by Spearman correlations. Here, the E. coli was the major host of the highest number of antibiotic-resistant genes for the all groups. And this suggests that the E. coli is the potential contributor in increasing the antibiotic-resistant genes load. That's why we performed strain analysis for E. coli. So you can see here that the groups who didn't receive antibiotics carry the lowest number of E. coli strain across the study. If you look here at the VP1, who received antibiotics, so this group actually carry the highest number of E. coli strain across the study, except for the third time point. I hope you remember from the beginning of my presentation that those preterm babies, extremely preterm babies, received antibiotics for longer period of time as compared to the first subgroup or first subset of the very preterm babies. So here maybe because of the probiotic supplementation, which might eradicate some E. coli strain, this group carry lower number of E. coli strain as compared to the VP1 across the study, except for the third time point. So this actually suggests that the antibiotic-naive group have less E. coli strain, and probiotics might eradicate some E. coli strain. Even the infants receive probiotic for a longer period of time. So to conclude, gestational age is a major factor in the microbiota development. Probiotics administration render the extremely preterm microbiota composition and resistome composition similar to full-term healthy babies. Extremely preterm infants have higher microbiota maturity level as compared to very preterm neonates. Hospitalization potentially aggravates the antimicrobial resistance by increasing the antibiotic-resistant genes abundance. Fortunately, we have here probiotics that can alleviate the antimicrobial resistance by decreasing the antibiotic-resistant genes abundance. E. coli is a potential contributor in increasing the antibiotic-resistant genes load. Finally, further analysis is needed to assure the role of probiotics in horizontal gene transfer of antibiotic-resistant genes. If you are interested to read more about our paper, I invite you all to scan this QR code or just take a screenshot or something like that to read our paper. And I'm very happy to receive a feedback or comments or questions from you. And finally, I would like to thank my supervisor, Veronica Peterson, and Klaus Klingenberg, who gave us opportunity to work with this data. And a special thank to my research group, the host microbe interaction at the Arctic University of Norway, and CANS, who funded this project. Thanks so much for your attention.